cash crop soybeans account for the largest source currently used today for animal protein feed. It is also the second largest source we use for vegetable oil. Worldwide, the U.S. is the leader in soybean production and export. To put into perspective, according to the USDA statistics report for 2011, the amount of soybean committed farm acreage was at 75 million acres, which made a net financial return on investment of $36.6 million. Of those 75 million acres, the state of Minnesota planted soybean on 7.1 million acres, roughly 10% of the nation's production. These acres break down to around 44 bushels per acre that net approximately $11.94 per bushel. Total net gain adds up to around $525.36 per acre. According to the Journal of Nematology, the statistical loss of soybean yield for the state of Minnesota in 2005 from root rot caused by Fusarium strains was a reported 84,902 metric tons. This disease state is called sudden death syndrome, SDS, and was a loss of around $20 million in 2005. This was the last reported efforts from many scientists around the nation to quantify such a loss due to SDS. Considering the demand locally and globally for soybeans and their products, we must raise the question, how do we develop an environmentally sustainable solution to prevent the future loss of soybean crops? Currently under investigation in a parallel study are the possible applications of secondary metabolites from endophytes, which are already found within the soil in which soybeans are grown in the state of Minnesota. In conjunction with the baseline deficits reported by this project, together our goal is to answer the question of how to design a treatment in which timing could be an integral factor in producing positive results. To establish the most effective timing schedule, our experiment involved the measure of photosynthetic activity throughout the life cycle of the soybean plant while infected with multiple known pathogens. To record a set of measurements, we incorporated the following methods. They began with the following materials: 35 pots, which were 4.5-inch planters, 105 soybean seeds, and multiple varieties of sorghum, both treated with and without pathogens. We first planted using a general potting soil in the controls. We used one set of 5 reps containing just potting soil. Another control was also planted with non-treated sorghum. Each consisted of 5 plants or reps. We then used treated sorghum with Solani, which were coated with 3 specific strains of fungi commonly found in Minnesota soils: Fusarium oxysporum, Fusarium solani, and Fusarium virgome, along with a pathogenic or disease-causing bacteria also found in Minnesota soils, Rhizoctonia. This coated sorghum was mixed into the soil, and these variants also had 5 reps per treatment. The environment in which the experiment was conducted was semi-controlled in a campus laboratory using metal halide growing lights on a 24-hour light cycle. The placement of plants was also along a windowed wall, which was blocked by a sunscreen. They were watered daily except on weekends. The seeds were planted in excess to allow for the predicted failure rate of around 10% in the successful germination of soybeans. And in the 3rd week, we selected the most suitable plant per pot and disposed of the extras. At the 3-week point, we also obtained our first data set. Using the portable photosynthesis system, we measured multiple parameters. By mimicking the precise environmental conditions the soybeans plants were growing in, we could measure the photosynthetic rate based off the input we used to mimic those conditions. The following variables were then controlled within a small chamber in which we could insert a small portion of the leaf to measure that rate. CO2 was controlled by applying an agent that removed excess CO2 from the surroundings. At the same time, CO2 was pumped into the chamber from a controlled source so that we could maintain that specified level. We also controlled flow rate, which would mimic the amount of wind around the leaf surface area. This would affect a small area at the surface of the leaf that affects the rate in which the leaf will exchange CO2 and O2 through the stoma. In conjunction with controlling the relative humidity via the application of a desiccant, we would maintain the environment conditions of our semi-controlled laboratory setting. Now that these variables were accounted for, we were then able to set the leaf within the chamber. Since this chamber was isolated, we were able to gather the photosynthetic rate the leaf was currently undergoing. Photosynthetic activity was then recorded, which is a measure of CO2 fixed or converted to sugar per leaf area. This method has been used many times for varying purposes, and with the exception of using the portable photosynthesis system as an instrument within the last year, many times here at Augsburg College as well. In an analysis of the preliminary results, we can already identify lower photosynthetic activity across all treatments with the exception of the variant applied with the Fusarium oxysporum strain. As to why this might be, discussion brought to light the possibility that the presence of this fungal strain may induce a response in which the soybean plant increases its rate to compensate for a deficit that the fungus has to it, whether it be in its stores or some factor along the metabolic pathway of photosynthesis. Another anomaly present is the treatment in which non-pathogenic sorghum was mixed in with the soil. Considering it has produced the lowest levels of photosynthesis, we can only apply some error as the cause to this variance results. The error could be from the investigators placing them in the pore-slit portions of the growth system, not supplying the 5 reps with enough nutrition to germinate properly, or the least likely in which they were all unfit individuals of soybean plants. Only repeated experiments may provide an answer to these two anomalies. At this time, the data so far is far from robust enough to generate any real conclusions that may or may not establish a firm timeline to identify exactly when soybean plants begin showing deficiency in photosynthetic activity. Even though some plants are showing physical symptoms of the disease states, without further investigation, we're not able to develop the timeline as of yet. Our methods are still prone to investigator error, and only repetition of the experiment will produce viable results to then combine with parallel treatment such as the one discussed earlier to develop a means in which we can combat these pathogens in an environmentally sustainable manner. Preliminary results from this first trial suggest that this path of investigation is very promising and that in the very least, everyone involved may contribute to a future solution. Thank you.